Hi, everyone. So I'm Janis. I'm a fourth-year PhD student working with DocEdge at USC. And today I'm going to share with you some interesting details about genetic and phenotypic comparisons. As evolutionary biologists, we are often interested in comparing genetic and phenotypic differentiation. And in some cases, we want to compare genetic and phenotypic differentiation because we want to learn about the action of natural selection. An early case was in 1949 when civil rights developed those F statistics, and people used it to learn the color variation of Linesense perrier, a native plant flower, and Mojave Desert, which is located in Southern California. And QST-FST comparisons has been a major framework for comparing those differentiations. And we often rely on FST, also a statistic due to civil rights, which measures the genetic differentiation. And for measuring phenotypic differentiation, we rely on another statistic, which is an analog of FST called QST. It measures phenotypic differentiation. And FST is usually known as, we can use it to learn about the null distribution of QST because we can use FST to learn the shared evolutionary history among different populations. And in expectation, QST should be equal to FST under neutrality. And in the past 30 years, this has been a very popular framework in common garden studies. If QST is much larger than FST, then that is consistent with selection driving phenotypes among populations apart. And if QST is much smaller than FST, that is consistent with stabilizing selections on traits with a shared optimum value. This framework has some ambiguity though, because there are different measures of FST. Which one should we use? So a very intuitive way that people often use to calculate FST is the locus level FST. So here we show how we express the locus level FST of locus K. And that is the heterozygosity among subpopulations versus the total heterozygosity. We can also notate them as VBK to VBK, that is the ratio of genetic variance between subpopulations to total genetic variance. And after that, to compute an overall FST, we have to grab information across all the loci measured in the loci that we've been targeted. And a very intuitive way is to calculate FST via an average of ratios approach, which is that we just take the average of all the FSTs measured across the loci. And another approach is to measure FST via a ratio of averages way. That is, we compute an overall FST by calculating the mean of the numerator and the mean of the denominator separately, and then take the ratio of those two components, and then we can have a ratio of averages FST. The choice may seem arbitrary, but it matters. In empirical studies, average of ratios estimators give lower estimates of FST. So here's an example in human data. When we compute FST from the 1,000 genome datasets, which is sequence data, we can see that there is a factor of 2 difference between these two different approaches computing FST. And the estimates from average of ratios estimators are exactly lower. This difference is not as pronounced when we are looking at the data computed from HapMap3, because those are HapMap3 data is based on common variance mostly, but the difference is not negligible either. So now we have many ways to compute an overall FST or to compute a FST distribution. How should we choose the FST distribution as a null for the QST-FST comparisons? One common recommendation is that we can use the distribution of single locus FST, and this has been proved to be a very good null when you are comparing the FST and QST in the trait only affected by a single locus. another way to choose the null is that we can use the scaled chi-square distribution, also known as the Lewontin-Quirker distribution. And it's often parameterized by average ratios FST in previous literatures. And also they recommended it using this kind of null with the single locus trait. But on the other hand, we can also use the scaled Lewontin-Quirker distribution parameterized by ratio of averages FST estimators. So here comes the question, which forms of FST and QST should we compare to test the hypothesis about the evolution of complex traits? That is, what should we use as a null distribution for QST? Should we use the distribution of single locus FST? Or should we use the Lewontin-Quirker distribution parameterized by average of ratios FST? Or should we use the Lewontin-Quirker distribution parameterized by ratio of averages FST? I carry out simulations to find an answer to those questions. So first, I simulate various demographic models under neutrality. And next, I generate an approximate joint allele frequency spectrum according to those coalescence simulations using the branch lengths to infer the allele frequencies. And next, I generate many random variants to calculate those different kinds of FST estimates and then compare them to the non-equilibrium FST. And finally, I generate random effect sizes and assign them to those variants to create different trait values and then compute QST distributions from those values. So here I'm going to show you the results of the distribution of single locus FST and of the single locus trait QST and also polygenic trait QST. So here, let's first look at the distribution of single locus FST. So we can see that the curve of the Lewontin-Quirker distribution parameterized by the average of ratios FST is a better match to the FST distribution, while the ratio of averages FST shows a lower peak and a longer tail. It is also similar when we are looking at the distribution for a neutral single locus trait, QST. When we are looking at this distribution, we know that QST is essentially equal to FST when we are talking about the single locus trait. So it is very recommended to use the locus level FST and also the average of ratios parameterized Lewontin-Quirker distributions as a null for those FST QST comparisons. But things changed when we are looking at those polygenic traits. When traits become highly polygenic, we know that the QST values, the expected QST values does not change with the number of causal loci involved in a trait, but the distribution does. So when we increase the number of causal loci from 10 to 100, then to 1,000, we can know that the distribution of QST moves to the right, and now it is a better match of the Lewontin-Quirker distribution parameterized by the ratio of averages FST. You can see that from this distribution, the tail of the QST is perfectly covered by the Lewontin-Quirker distribution parameterized by the ratio of averages curve, and the two other curves are very strong and right skewed, so they are not a perfect match when we want to find a null distribution in those kind of comparisons. So why would this happen? Well, if you look back to the theoretical literatures on QST computations, you can find that the rationale lies in average coalescence times. There are many literatures pointing to that direction, and one informative one was from Mike Whitlock in 1999. And he used a coalescence approach to prove that the QST should be equal to Slatkin's coalescence FST. So those two terms should be inconsistent with each other under the coalescence interpretation. There are also some other good literatures supporting this theory. For example, like Edge and Rosenberg from 2015, and one from Koch in 2019. But a very strong one on theory is from Guerra and Nelson in 2022. And they found that the average of ratios FST estimators is essentially lower than the estimates. So it is not only shown in the empirical studies, but also proved in the theoretical studies. And so, as I just mentioned before, Whitlock study is not the only one that points to this direction. And the Adrian-Rosenberg paper found out that the expectation of QST is capped from above by the ratios of averages FST. And it is exactly lower than that for trade controlled by field loci. And Koch, again, using another approach, proved that for distributions, for Lewontin-Quirker null distributions, it is a good approximation for demographic models without spatial structures, like here for the symmetrical island model. So now I hopefully have convinced you with both simulations and the previous theoretical works. What does it mean for empirical studies? Well, type 1 error rates of QST-FST comparisons depend on genetic architectures. So you can see that using the Lewontin-Quirker distribution parameterized by average ratios, FSTs, gives a very high type 1 error rate. For this case, it shows about 0.75. And for some cases in our simulation, the type 1 error rate can go up to 1. While for using the two other, when we are using the two other FST distributions, for example, the locus level FST distribution is very conservative when we are looking at a single locus trait. But for the other, for the polygenic traits, it is not as good as the Lewontin-Quirker distribution parameterized by the ratio of averages FST. And we also want to mention that the ratio of averages FST estimators is under and or below or at the threshold in all the regions that we have tested. So before we have already talked about demographic models without spatial structures or with only modest spatial structures. What if we look at the demographic models with spatial structures? So now you can see that the Lewontin-Quirker distribution parameterized by ratio of averages FSTs is very strong, and that peak is very high, and it's not a good match with the QST distribution anymore in this case. But when we look at the multivariate normal distribution proposed by Koch in the Koch paper that we mentioned before, it provides a type 1 error rate. It produces type 1 error rates always at or below the threshold at the nominal rate of 0.05. And it is also similar when we are looking at the FST distribution, single locus FST distribution, but with common variance only. So it shows the same pattern when we are looking at their type 1 error rates as the Koch's multivariate distributions, multivariate normal distributions. This result makes sense because for sufficient common variance and sufficient short time of drift, it is okay to model the allele frequencies under a multivariate normal distribution to show to mimic the distribution of FST so that we can use it as a null with QST. And there's more to say than that because we also found that QST and FST estimators also have to be consistent with each other and under coalescent times. So here are two different kinds of FST estimators. One is from Ney and the other is from Weir and Cockerham. So they are different in calculating the average coalescent time to the most common recent common ancestors from two random alleles drawn from the total population. So this difference makes them pair with different types of QST. So FST Ney is a good pair with QST RB, and FST Weir and Cockerham is a good pair with QST PBS. So here we can see from the result that when we are looking at the results from the Lewontin-Quirker distribution parameterized by the ratio of averages Ney FST estimator, it shows a very conservative and calibrated type 1 error rate comparing with the QST-RB. So we want to just let everyone know that when we are choosing the right estimators, it is not like which version you choose is not that matter, but which versions you choose to match with each other, like the interpretation within the correlation time of the QST-FST estimators should match each other. So here comes to our conclusion. Compatibility of FST and QST in correlation times is more important than the choice of forms. And a ratio of averages FST is generally superior and reduces false positive rates. Yes, ratio of average FST is also closer to the true FST that we design in the simulation models. And if we are looking for empirical studies, how should we find a good one to follow? We can look at how they choose a null distribution for the QST-FST comparisons in their paper. So the first one is that if they use Lewontin-Quirker approach, it often works well, but it can fail with many themes that have strong spatial structure. And the second point is that using the distribution of single locus FST estimate can also be effective. And that is also what we often see in those empirical papers. But those only work for traits that are polygenic. When traits are polygenic, only common variance should be used to avoid high false positive rates. And I guess that's what most people do when they are doing empirical studies, like in common garden studies. So that's also our conclusion is also matching with their findings. And the third one is that if it is possible to estimate the necessary average within and among population correlation times well, then Koch distribution appears well calibrated for use with QST-RB. Which means that Koch's approximation of FST's null distribution, it's kind of like the FST developed by Nate. So we should use his distribution with the QST-RB. So that is the end of the talk. If you want more details of this guidance of QST-FST comparisons, you can find our paper in genetics. It's recently published. And thank you for your attention for the talk. And thanks for the support from my lab. And I'm happy to take any questions.
Great question. So is there any time I ever want to actually use the average of ratios? I feel like, or is the ratio of averages just always superior? Just repeat the question for the people in the back.
Okay, cool. So the question is there any time that we can use the average ratio of FST? Because in the talk, it seems that ratio of average FST seems to be always better than the other one. The answer is yes. If you're comparing the QST with only common variance included in the breeding values, then you can use the average ratios FST because that will not make a big change between these two kind of estimates. Yeah.
So how robust are these approaches if the patches are not homogeneous, right? It seems like you have to assume the patches are the same size and that migration between them is equal. What if the world had one giant patch and a bunch of little islands around it? Do you run into trouble with the averaging?
Yeah, that's a good question. So in my model, we did assume the mutual migration between every theme. So that's very naive. But in reality, we found that according to our search for different studies, you can still use ratio of averages. Like that's the better one because it's like a weighted averages for different locus. So that's a better strategy than do you use the very naive mean, which is the average ratios estimates.
Okay, last question.
It's a very naive question, but I was just wondering if I'm not mistaken, like the spatial structure is not fluctuating in time in your models. I was wondering if population structure varies, can it have an influence on all these estimates?
Yeah, great question. So population structure, I mean, like spatial, definitely has the impact on your estimates of whether QST or FST. Like it depends on your case, like it should be a case-to-case question. And for the time scaled population structure, I didn't look into that question, but I can look for some literature for you after the talk. And if you want to talk, we can talk later. Thank you.
Very good.